Upstate Medical University in Syracuse, New York invites you to be the informed patient with the podcast that features experts from Central New York's only academic medical center. I'm your host, Amber Smith. Wouldn't it be cool if, when disease or injury damaged the retina in our eyes, doctors had a way to replace the neurons and restore our sight? A vision researcher and his team at Upstate are studying regeneration and protection of the nervous system, and he's here to tell us about it. Levi Todd is an assistant professor in Upstate's Center for Vision Research. Welcome to The Informed Patient, Dr. Todd.
Hi, Amber. Yeah, thanks for having me.
When you talk about regeneration and protection of the nervous system, are you talking about growing new neurons or nerve cells?
What we're trying to do is protect the brain or the spinal cord or the cells in the eye from dying, because once they die, they're gone forever. So once you have an injury that makes you not able to walk again or not able to see again, those things are irreversible because those cells do not come back. Once they're developed when you're a young infant, that's it. So what we're trying to do is find different strategies to protect those cells from dying in the first place, or then after they die, if we can find different types of therapeutic options to replace those cells, because right now there really isn't any options after the cells are gone.
So, when a particular cell dies, I understand it's not coming back, but does our nervous system grow new neurons throughout our life?
It doesn't grow new neurons, but they can connect to new like partners. So basically, if you have one type of cell die, so for instance, there's a disease called glaucoma, that's a specific cell in the eye that connects to the brain that dies. Now, if that cell dies, you don't grow them back, but the surviving cells around it can kind of rewire. So that's called plasticity. So there's a little bit of plasticity in the nervous system, especially when you're young. So you notice like young children, when they have injuries, they're more resilient to recover. And for adults, they still have a little bit of plasticity, but it's way harder. So if you have a stroke and neurons die in your brain, you can go to occupational therapy and you can recover some of the, functions of, say, how your arm works again. But that's not because you got new neurons. It's just because you've strengthened the surviving connections.
Interesting. Now, are there mammals whose nerve cells do regenerate?
So that's kind of a complicated and controversial topic still. Uh, in the brain, most of the consensus is no. In the spinal cord, the consensus is no. Once those cells are gone, it's forever. But strangely, in the peripheral nervous system, so that means outside of your brain and spinal cord, the ones that connect, say, the nerves in your fingers, those can regenerate. And you've probably heard of like people that have lost sensation in their finger after a burn, and then maybe months later it's back. So there's this mystery about why do nerve cells regenerate in some parts of the body, like in your periphery, but not in the central nervous system, your brain and your spinal cord and your eye. So that's kind of a mystery within mammals.
Are they different nerve cells, the ones that are working in the brain versus the spinal cord versus the periphery?
Yeah, there's different nerve cells everywhere. So if you look just inside of your eye, there's hundreds of types. So when you're developing the nerves in your eye, there's types of neurons that only respond to light, some that only respond to motion, some that only respond to if the light's bright or low. So there are literally millions of like kinds of neurons, and they're all specialized to certain functions. So yes, the types of neurons that are in your periphery are different kinds than they would be in your brain or your retina.
So there's neurons that work on vision in the retina.
Yeah, exactly. So there's neurons that catch light, and then there's neurons that talk to the cells that catch light, and then there are neurons that go from your eye back to your brain. So those are the ones that die during like glaucoma. When you lose those neurons, there's no way for your eye to talk to your brain anymore. So that's why people go blind, and that's why it's forever.
Now, remind us, where in the eye is the retina?
The retina actually is part of your brain. So when you were embryonic developing as a human, there was part of your brain that literally bulged out to make your eye. So it's kind of wild. So there's a piece of your brain that literally floats outside of your skull. So in the retina where it lives, it's this thin lining that's on the back of your eye. So it's not in the front of your eye. If you poked your eye with a needle, you were not going to touch your retina. The retina is deep in the back. And it's a really, really thin layer of literally brain tissue that just kind of lies on the back of the eye. And it has all this complex like circuitry that senses light, talks to each other, and then sends it back to your brain for more, visual sensation.
Now, there's glial cells involved in some of this too, right?
Yeah, so glia cells were a mystery for a very long time. And in fact, the word glia is meant like glue in Greek. And they thought historically in neuroscience, these were just like passive concrete glue that just like kind of keeps your nervous system molded. But since the last like 20 or 30 years, this field has exploded, and these glia cells are basically the support cells of neurons. So neurons are the ones that are famous. They're the ones that have connections to each other. They're the ones that send electricity to each other. The glia do all of like the dirty work. They clean up all of the garbage that's in your brain. They clean up little pieces of neurons that break off. If things infect your brain like a virus, the glia are the ones that like protect you from that. So glia are basically the support cells, and they're the only reason neurons can kind of do their job because they have these like support staff. And that's what glia are.
If the nerve cells die, are the glial cells the ones that may kind of fill in for that cell?
Yeah, so that is really where our work kind of focuses on, because this story I was telling you about how neurons don't regenerate in mammals, that's not a rule across all of biology. There are species like fish and frogs and salamanders that can regenerate their brains, their retinas, their spinal cords. It's really amazing because if you look at a spinal cord of, say, a fish, it's not that different than a human. It has the same types of, columns of neurons and nerve cells. It connects to different motor functions. But if those neurons die in a zebrafish, just a species of fish, those neurons regrow and the fish can swim again. Or if those neurons die in the retina of a fish or a salamander, those neurons can regenerate. That was known since the 70s. You knew that if a fish went blind, it can see again, but people didn't know how this works. It was a big mystery in regenerative biology. And the way that that works is these support cells, these glia cells, they can sense that the neurons are lost and do something called reprogramming. But what that means is you lose your identity. You used to be a glia cell, you stop being a glia cell, you become basically a stem cell, and you can regenerate neurons. So these glia cells in some species can be a source for new neurons. So what we're trying to understand is, can we do that in humans or in mammals like mice?
This is Upstate's The Informed Patient podcast. I'm your host, Amber Smith. I'm talking with Levi Todd, who's an assistant professor in Upstate's Center for Vision Research. Can you explain the two ways that your lab is exploring to get the neurons in the retina to regenerate?
I already mentioned here, when neurons are gone, they're gone forever. And what that means then is there's really no therapeutic options. So what we want to do is something called cell replacement strategies. So basically, if you're late stage in disease and you go to the doctor and you have glaucoma and they're like, hey, all of your ganglion cells are now gone, we're sorry, we can't do anything. So we are really at that point. We want to see, what can you do once the neurons are gone? So what we do is we focus on these cells that are called glia, and these are cells that are already in your eye. They're the ones that do all the support functions I already mentioned for like neuronal health. And what we want to do is reprogram them. So basically, can you make these glia already in your eye into basically stem cells to regenerate new neurons? So the way we do that now is we use mouse models and some human models where we try to introduce genes inside of glia that can make them basically into stem cells again. So there are these famous genes in development that make stem cells into neurons. What we want to do is reintroduce those, force those back into adult glia, and see if they can become basically a source for this rejuvenation, these new neurons. And we've had really good success doing that in mice, which is a really good first step because mice are mammals. They have a similar genome to humans. So we think if we can understand kind of the genes that could control the generation of new neurons in mice, we can start trying to do that in humans. And we started doing that in some types of like in vitro, but they're like plastic dishes basically, where you can have human cells growing in dishes. We can see if those same genes we're discovering in mice can then be forced into human glia to make glia into neurons. And then hopefully the future then is that, can you introduce those into adult humans and then maybe be able to restore vision? So those are the like angles that we're focusing on right now for restoration of sight by giving new neurons back to the retina.
So the work would be done in the lab and then you would inject it back into the patient.
Yeah, so our lab's a basic science lab. So what we mostly do is try to discover the fundamental mechanisms of how certain genes can control the ability of glia to make neurons. Now, when you want to scale us up to humans, it takes a lot of safety trials. It takes a lot of like animal testing and large animal testing. So what usually happens is we try to discover certain cocktails of genes or different treatment therapies that look like they might work in animal models. And then what happens usually is companies come in and they say, hey, let us see your data. And they try to develop more therapeutic avenues. This has already happened to some of our work. So when the bigger companies come in, then they try to scale it up. They try to scale it up. How do you give this to a human? Will it be safe? How long will it take? How much money will it take? How many years will it take? So our lab really is at the ground floor of this, but we've already had success in company interest of like starting to develop therapeutic options.
So the success that you've seen, like in mice, have you been able to restore sight?
That's a hard question because mice don't really care about their vision. Mice are like nocturnal creatures. They use their whiskers. So actually to tell if a mouse can see or not is actually not so simple. The short answer is no. We don't know if it can restore vision or not, but we do know that the new neurons can connect into the circuitry. So what that means is that we've been able to generate new neurons in the adult retina. We can look at them with the genomics tools we have, with the imaging tools we have. They look like real neurons. They act like real neurons. We can do things where we can physiologically record from them. So you can put basically an electrode into a cell, you can shine light on it and see if it responds. What that means is that it's functional. It connects to partners. So that's really exciting to think that it could do something good for vision, but we've yet to get to the scale of like understanding if we can make a blind animal see again. But that is really in short order for us. Like that's in our current aims of our lab.
And what about the body's immune system? Does that have a role in vision, and would it impact any of the work that you're doing?
Yeah, so that's a great question and also a huge angle of our laboratory now. So traditionally, people said there was something called the blood-brain barrier. And what that meant was the immune system stays out of the brain because the immune system is like an army. They're looking for viruses or bacteria. They get upset and angry and they eat it and they just like release things that cause destruction because usually you want to kill invaders to your body. It's okay when you're in your skin or you're in your stomach because those cells can repair themselves. But in your brain, if you go overboard with the immune system, you're going to be out of luck because you're going to damage cells that can't be replaced. So traditionally, there was this barrier that kept the immune system out of the brain and in the eye. Now, what we've been finding as a field in neuroscience and immunology is that this breaks down in disease and in aging. So diseases like Alzheimer's or Parkinson's, which result in the loss of neurons, you see a swarm of immune cells from your peripheral body, it's usually not in your brain, are now in your brain. It's the same in the eye. So these famous diseases like glaucoma or diabetic retinopathy or regeneration, there's a huge immune component here. And what happens is cells start to die. That causes this inflammation. The peripheral system now invades where they shouldn't be. And that can cause this runaway effect where more inflammation causes more neuronal cell death and that exacerbates degeneration. So we're really trying to understand now what keeps the brain and the eye healthy normally, but when it goes wrong, why does the immune system invade in? And if it does invade in, what can we do to stop it from going overboard, from doing more damage than good? And that's a really big angle of our lab right now.
Well, you've got a lot of important work underway. Thank you so much for making time to tell
Thanks.
us about it.
Yeah, thanks so much. It was really fun.
My guest has been Levi Todd, an assistant professor in the Center for Vision Research at Upstate. The Informed Patient is a podcast covering health, science, and medicine, brought to you by Upstate Medical University in Syracuse, New York, and produced by Jim Howe, with sound engineering by Bill Brokel and graphic design by Dan Cameron. Find our archive of previous episodes at upstate.edu/informed. If you enjoyed this episode, please invite a friend to listen. You can also rate and review the Informed Patient podcast on Spotify, Apple Podcasts, YouTube, or wherever you tune in. This is your host, Amber Smith, thanking you for listening.